Welcome to day 2 of the TERN Science Symposium. It's great to see you all back here and getting into the spirit of things by arriving early, because we've got a fantastic lineup for you today. I want to tell you a little story first about Brisbane, which was founded on the homelands of the Turrbal and the Jagera people. And so before European colonisation, the area of Brisbane was called the Meanjin, and that was a name by the Turrbal people. And Meanjin, interestingly, means a place like a spike. And that, if you're a visitor to Brisbane and you've had a look around, it's a reference to that spike of land where the Brisbane Central Business District is now located and shaped by the shape of the Brisbane River. So TERN acknowledges this country and its traditional custodians. We acknowledge and respect the spiritual relationships between the traditional custodians and this country, which has inspired language and songs and dances and a body of traditions and knowledge and dreaming stories over many thousands of years, and which we are starting to learn more about. So we pay our respects to the elders, those who've passed, those who are here today, and those of tomorrow. And it's my hope that we continue to peacefully walk together and in respect and kindness in caring for this country and each other. So I'm now given the great pleasure of introducing our plenary speaker for day 2, Professor Bronwyn Harch. Professor Harch, Bronwyn, is very, very special to TERN. She was a member of our advisory board when we were in our early days, and so if you're enjoying the TERN Symposium, you have to say that Bronwyn had a little bit to do with the direction in which we have been able to go. There's a very long biography of Bronwyn, which I encourage you to read because it's inspiring. But importantly for us in this symposium is that Bronwyn has been a leader in research and innovation strategy, with a passion for brokering the transdisciplinary area, not just between private and public alliances, but across a whole range of different science domains. And I think our Science Symposium is showing that if we're going to be facing the ways in which we address the complex societal challenges of this century, that we need that transdisciplinary approach. We need to be at the borders. So please come and tell us a little bit more, Bronwyn. Thank you.
So the advertisement of the organisation I work for, Griffith University, happens in October. So for those of you wondering, it says QUT here. I started at Griffith University in October as their Vice President of Industry and External Engagement. So what I've been asked to speak about today is creating alignment for environmental information impact. And the journey I'll take you on is a little bit about me to give you the context of the way that I'll be speaking about things, speak about the many dimensions of environmental information, and what achieving alignment through this transdisciplinary kind of practice that Beryl's been speaking about, what it means from my experience. And then it's a little bit, we'll get a little bit HR actually, where I'll speak a bit about reflections on why it does often hurt, while you feel elated, while you feel frustrated when you're trying to achieve alignment for environmental information. So I'll reflect a little bit on strategies there. So I also wanted to give a special shout out to all of those people online, being very committed and coming in online. I'm delighted that you're here with us this morning as well. So in terms of my background, I'm a data scientist that studied at Griffith University, actually environmental science, and then I did my PhD, which was looking at how you put together all these three things: agricultural science, data science, and environmental science. And the picture on the far left is a picture of peanuts. So when I was doing my PhD, I worked for peanuts. So I was working for the peanut industry, looking at large and complex plant breeding, germplasm datasets, if you know about gene banks, looking at the many different kinds of space and timescale information they were collecting, and how do you put all the different information together. So that kind of from very early on, I've always been very motivated by the industry problem and how to help them practically. And in terms of the kind of journey I've been on, I wanted to kind of take a little bit of a moment to talk about journeys, because all of you are on a different dimension when it comes to thinking about your journey, whether you're in a research organisation or somewhere else. And I often depict a journey, well, maybe it's my journey, in this pedagogy of a zig and zag. And as you went from PhD to postdoc to independent scientist to leading teams of quants, so it wasn't just all the data scientists, statisticians, it was all the other people doing mathematical modelling and IT and visualisation, and then leading multidisciplinary teams where you've got to also respect all the agri-domain kind of knowledge. I know Mike Grundy's in the audience and taught me all I know about soils, for example. Setting research directions, how you then have to start seeing patterns and thinking about where research directions for your group or your organisation go. And then most recently, I've been working as the interim Queensland Chief Scientist and thinking about how research and science policy is set for the state, and of course how you interact nationally and globally in trying to achieve things for the environment. And I think for me, TERN is this amazing vehicle that has progressed so many careers. Beryl, it'll be interesting to see how people's careers have changed and been impacted and positively influenced through the leadership opportunities that TERN has. You might want to kind of track that. The other is the forums that many of you get to globally because of your engagement with TERN as a national research facility as well. So I'll move on to now the many dimensions of environmental information, and the reason I'm on day 2 is because of the focus, particularly in the next few sessions, around environmental information and the analytics kind of dimension around it. So this is the way I kind of think about the world in terms of information and decision-making value chains. So there was a period of time I was fortunate enough to be in CSIRO, and I led what is now called Data 61 in CSIRO. It was called something else at the time. And we often thought about these information and decision-making value chains, which are sector agnostic, but the different dimensions within it are very important. So if I start at the top, designing for data generation and capture, these things are so easy to speak about, but the depth of capability and knowledge and experiences that needed in each one of these, as we go around the circle, is very deep and an absolutely amazing opportunity for you to learn within each area, but across. And I expect for many of you, you do your work on a number of arcs in this circle. And I was delighted when I was speaking to Mark Grant recently to hear about the data protocols that TERN has been leading for the country as well. So in that designing for data generation and capture, it's not only the space-time concept, of course, but it's how you're going to capture it, what's the technology that you're going to use. And I know there's a lot of experimental technology that TERN gets to experiment with as well. Moving, of course, through to data generations, communications and capture, and boy, has the world changed in terms of what we're able to do here. And I think this is the great part about the citizen science component as well. I got exposed a lot more to citizen science as the interim Queensland Chief Scientist, and how the different communications and capture really are fueling a lot of the citizen science that we're seeing as well. The kind of part of the ARC that I spent a lot of my time in was integration of data and modelling, and I was very excited to see, particularly at the bench kind of career, that hybrid modelling that's so rife now, where it's not statistical modelling or deterministic modelling. There's a lot of melding in hybrid models these days, and I know a lot of you would be engaged in that kind of context. Of course, a big component for TERN is making sure there's the storage, discovery, communications and access. Again, very easy to say, but to really have the open kind of context and the way people can access things are really critical. And I know that through the Nature Positive Plan that the federal government, that Tenure Plebiscite's department, that part of DECWI that Tenure Plebiscite looks for, there's establishing environmental information as it's like a BOM-like entity, won't be like BOM exactly, but that kind of entity. So I think it's going to be very interesting for TERN to think about when it's developing data protocols, the kind of access to data, how a lot of the learnings that TERN's already had can be brought to bear for the Nature Positive Plan and that environmental information entity that will be born. Importantly, it all comes down to gaining those insights and understanding, and there's a few talks I saw yesterday morning where people are going around the arc a little bit more in terms of making sure that the visualisation and that interaction with information for end users, whether it's in government or in industry or the community groups, that they can actually gain the insight. It's not just the report, there's visualisation means and immersive kind of analytics that people are using with environmental information as well. Decision making under uncertainty, and this is making sure that that uncertainty element is there, and as a statistician you'd all expect me to say that, I'm sure. Then how you then go through, once you've set up that information value chain for a particular purpose, how it actually gets implemented operationally, and then how you adjust that as well. So that's kind of the way I think about these information and decision-making value chains, and that they are certainly sector agnostic and a big component of what TERN's trying to do to really fuel that these circles go as easily as possible. And that TERN is often providing one of the key data sources, or many, for any particular value flow. And so I mentioned before about the Nature Positive Plan, and when you're thinking about what they're trying to achieve there, for me this kind of graphic, particularly the stuff in the middle, is what they're trying to achieve for the modelling of the key processes, the next generation data acquisition technologies, and great to see that Australia's investing in the space-related technologies. I know TERN has a really good relationship with the various different space-focused organisations that are developing. That integration and synthesis of observational data and that risk-informed decision-making, where the thing in the middle is, how do we have information that's going to inform environmental accounts? How is it going to inform landscape stewardship, environmental services, community wellbeing, as well as a competitive edge for the country? I think that's what both Hugh and Andrew was speaking to yesterday, was about that middle bit of what the real impact of the information we're collecting environmentally. So the next is around achieving alignment, knowing you have these kind of information value chains and you're trying to achieve alignment, and then in any value chain there are many actors in trying to achieve the insight that's needed. So what does it mean from my experience? So the biggest opportunity that I see for any research and innovation ecosystem is how you achieve alignment. And the actual birth of TERN and many of the NCRIS facilities came through alignment of people about what was actually needed. So this is TERN's kind of living the dream in terms of this alignment piece. But in a research and innovation ecosystem, people often speak about research and industry, and if only it was that simple. And I often depict it with these 5 different actors of government, corporates, startups and SMEs, research organisation and capital investors. And I've depicted government in terms of different coloured dots because no one government department is the same, whether it's federal or state. And likewise for the other kind of entities. And so as a research organisation, I've put you there, Beryl, what's the role of an organisation like TERN when you're trying to achieve alignment? And I know there's a lot of work that you do to achieve alignment with these different entities. I think increasingly we're seeing that there would be a lot of startups and SMEs that are relying on increasingly NCRIS kind of facilities in terms of their information. I know there's a few startups that I interact with that work with quite a number of the NCRIS facilities. And so how do all of you have a deeper relationship through their intelligence that they have from their market of creating new value propositions for them as well? Maybe some of you are even thinking about founding particular entities as well. I was speaking with someone, David Swain, if any of you know him from up north, and he's established a startup which is about serving analytics capability in the cloud to bring in information. So there's a lot of different opportunities for us as a community, but also for others engaging with us around environmental information for that alignment piece. So aligning to what? And so before I get to the what, I want to speak through to these three kind of elements. And if any of you were seeing me speak as Interim Queensland Chief Scientist, this is me still banging on about the same kind of things. At least I'm consistent. And so it's around these foundational capabilities of the talented researchers or the talented people in the system, the leading edge infrastructure for research and for translation, and that's squarely where TERN's fitting in, accessing the talented researchers, accessing the translation partnerships. It's a key piece of KPI for TERN, is making sure those partnerships are solid and people are saying it's really valued back to their investors, and also having an engaged community. But in relation to achieving alignment, and the things that I've put there as research missions and deep tech enablers aren't specific to TERN, but the idea is, what are the missions you're trying to work towards? What are the major problems that you're actually trying to solve? And I know that TERN has a number that it's facing. So I often think about things of sovereign capability, because you've got to think about where the money's coming from. So using the term sovereign is probably a good idea. The economic advantage and also societal resilience. And so many may think straight away with the societal resilience that I would say TERN would be in protected, restored and sustainably managed ecosystems. But of course, it's a lot broader than societal resilience. It's equally important in sustainable agri-food systems, in transitioning to a decarbonised economy, for example. So thinking through for TERN, what are those key missions, but also as you and your organisation, what are the key things that you're actually trying to solve for? The deep tech enablers, I often put these as equal pegging as missions, or whatever it is that you want to call them, core focus areas. Often people put them underneath as enabling and supporting, but as someone who comes from those disciplines, the thing that you often are torn between is, are you leading or are you supporting other people's endeavours? And what you have to try and do is strike that balance, whether you're running a whole outfit of stats and mathematicians, or you're actually with your career. And you often find that at different times, when you're in the deep tech enabler area, you're between often, and you have to figure out that balance that's right for you between leading and supporting. And there's a key component, of course, for TERN, that there's a lot of quants out there in terms of being those deep tech leaders. Another final concept that I wanted to speak to is around research and innovation. And this is something that I kind of was distressed a little about when I was working in government, that often when I was speaking to many people in those 5 areas of alignment, speaking about research and innovation, they went to the right-hand innovation funnel where startups are being kind of pumped out of the system. And I often had to get them to realise about their investment across discovery, application, demonstration and deployment of research. And kind of slowing down the dialogue about R&D, or research and innovation, that it has these different phases. And you also have different partners. So when you're doing research with your new knowledge, technologies and practices, it depends where you're at in that kind of discovery cycle, discovery, application, demonstration and deployment, the kind of partners that you have to get your information or your technology into the hands of someone. And what I've found is that people often miss this linkage piece, which how you achieve alignment to be able to then think about what's the things that should be going into that innovation funnel. So lastly, am I going for time right on? Just some of these reflections that I wanted to speak to. So again, this is the world according to me. So this has been my kind of experience. Learn a new language or two or three when it comes to these kind of transdisciplinary endeavours. Be very deliberate about the networking events you go to. Sometimes you do go for the content, other times you go for the people that are going to be there that you get a chance to speak to, particularly if you're breaking into new kind of networks. Make sure you listen with your eyes and your ears. You can tell a lot about whether people think your ideas are good or not by the eyes and the ears. Shared goals are really important, but you have to consider your different KPIs. That's where it's respecting that you can have a shared goal together, but what I want to achieve and what you want to achieve may be very different, but you can find the shared goal. When you're hunting in a pack, make sure you have the best with you. You can waste a lot of time if you don't have the best people with you. The next one, the importance is the pause bit, and it's about when you're asking end users directly about their challenges, make sure you pause. Can I help you with that? Pause. And how can we make this happen so you can co-develop? So it's that listening piece that's so important when you're speaking to end users. It's okay to say no, but you have to learn how to say no your way when it comes to being part of these transdisciplinary endeavours, because often when you're a transdisciplinary kind of person, you're very passionate about solving the problem, but you've got to make sure it's the right problem for you to be solving. Home straight now, Beryl, if I can get this to work. The relationship management is key. Never underestimate the amount of time that you need to put into relationship management. I think this is probably one of the most important lessons I had to learn in my mid kind of leadership career was no from an end user does not necessarily mean never. It's always about timing. So it could be no for now, particularly if you're working with government and industry in terms of their board meetings and all of those kind of things. It could be just not now. If it starts badly, it usually ends badly when it comes to a lot of large-scale kind of endeavours. So if it's feeling like that, probably walk away. Some days you win, most days you do learn, and that's a good thing. Feedback, it's about how you receive it, not about whether it's good or bad. It's how you respond to it that people will notice. When someone's telling your ideas, probably not that great. Don't be afraid of being different. Be afraid of being the same as everyone else. And as someone who was a statistician in a very large organisation that had so many different domain areas, it was often difficult to be heard because people thought you were different, but in the end that was my main value proposition as a statistician that knew a lot about the agri-environmental domain. And finally, I spoke about struggles, and I'm very thankful for all the struggles I had when I look back on it, because it was through those struggles that I figured out what my strengths actually were. So I wish you all the best for the rest of your deliberations today, and thanks for the opportunity to speak to you.
I'm very happy to ask. That was fantastic, wasn't it? And I know it resonated with me, so much of it, and I'm sure that all of you found personal reflections happening as you listen to Bronwyn. Does anyone have a question for Bronwyn before we finally thank her and farewell her? Any questions here or online? None online? Okay, well, thank you. Thank you, Bronwyn.
Thanks, everybody.
It's fabulous. And now I introduce the first of our session chairs for the day, Will Woodgate. Those who were here yesterday heard him speak, and I think you will agree that, having listened to him and look at the topic that he's going to be chairing, technology for conservation, artificial intelligence, machine learning, associated data challenges, Will's faced it all. He's a great person to chair, so I'll hand over to him. Thank you.
Thanks, Beryl. You've basically done my job to introduce the session. So we've got 6 wonderful speakers lined up today, including 2 online. In the spirit of keeping to time, just like yesterday, we'll have self-introductions. I refer the audience to the program with the bios if you want to read up more about our speakers. And then also please submit your questions via the app, so we'll have a question and answer at the end of the session. So without further ado, our first speaker is Kathryn Hall.
Oh, good morning, everyone. It's absolutely lovely to be here, and I was even more excited to hear Bronwyn talking about it's okay to be a little bit different, because when I put in the abstract to come and speak at TERN about the Australian Reference Genome Atlas project, I was a little bit worried that we were a genomics project, not an ecology project, and hearing her say it's okay to be different gives me complete license to go ahead and give you this presentation now. So, as I said, my name's Kathryn. I work at the Atlas of Living Australia, and it's my absolute pleasure and delight to be leading a fantastic team as we're putting together a pilot project called the Australian Reference Genome Atlas. And part of one of the things that we're trying to do with that is to integrate genomics data with existing biodiversity data that's held within the Atlas of Living Australia. As I said, I'm from the Atlas of Living Australia, but the team that I lead is also has members from the Australian Biocommons, and we're also proudly supported by Bioplatforms Australia in our endeavours. And we have some additional funding from the ARDC, and we will be producing an NCRIS-enabled platform throughout the project. So, I suppose the first question is, why on earth would we even be building this thing called ARGA? What's it all about? Well, I think it was born out of a bit of a recognition that genomics data are becoming increasingly inexpensive to produce. I was talking to a friend of mine just yesterday who said that she can get me a whole set of SNPs for $800 now. I was like, oh my goodness me, that's so cheap. And it's becoming cheaper and cheaper and cheaper. I know, it was $5 for a sequence, so that's cheap. But relatively, it's becoming less and less expensive, and there's more and more of it. And it's being hidden all over the place on the internet. And yesterday it was mentioned about people not really wanting to share data, or maybe not wanting, but perhaps having a reluctance. And that makes it very hard for researchers to actually know who's done what, where is it, and could they possibly even reuse it in their own analysis. These data can be placed into a number of different repositories. The chief amongst them is NCBI, but these repositories aren't necessarily interoperable with other repositories, so you search one, but you don't know if there's data relevant to your species or your question on another repository. So that becomes quite difficult, and you end up just collapsing in a heap and saying, I can't find anything, it's just too hard, I don't know, I'll just sequence it again myself, which is a problem because we're duplicating effort there when we could be investing it into other places where that money could be better spent. Now, one of the things that's most exciting about genomics data, of course, is that it can be leveraged for a number of different questions. And I suppose one of the most important things about it is that it allows us to then make perhaps slightly more evidence-based decisions about where we might want to introduce management controls and that sort of thing for threatened or vulnerable populations or environments. At the moment, there are about 15,000 life scientists who are trying to engage and deal with this burgeoning mass of genomics data that's out there. And because of that, we feel that creating something like ARGA could actually solve a big problem for quite a number of people and pretty much accelerate our efforts in that area. So, and also another point that Bronwyn made this morning was about bringing things into alignment, and I like to think that perhaps that's what at ARGA we're really doing is we're trying to align a lot of this genomics data, make it findable, and make it findable within context. And we're aligning it with the taxonomic data as well as some ecological data and phenotypic data where it's feasible to do so. I should say from the outset that we are not a repository. We are not taking in data, but we are simply indexing it and taking it back to the original source. So when people find it from us, we then link them back to where it came from in the first place. In brief, the way that our little rocket ship has been going is that we've been spending the last year designing what we're doing and how we're getting there. And we've just this last month released a UI for testing. We haven't loaded all of our data sets yet, but I would invite you to have a look at it, tell us what you think of it, and if it's a way in which you enjoy looking for data or it's actually useful to you. We will be releasing the full version in September, and at the end of the year, just after a few iterations based on the feedback from the community, sending it out there to the community for use. Very briefly, I just wanted to show you that we did actually go through a design process where we spoke to people and asked them about the sorts of data that would be relevant to them and the ways in which they would like to search for things in context. We've spoken so far with about 100 people from 38 institutions, and in the main they've come from biosciences or taxonomy and biodiversity domains, although we have spoken with people from agricultural sectors as well. The other thing about ARGA is that it works on a very complex data space. And I made this graphic. You don't need to worry about the detail. It's simply to show you that genomics aren't genomics. There's all sorts of different types of data sets that are out there. It must be exactly the same for you in the ecology community where, you know, you get all sorts of different data and they're not necessarily the same, and yet they've been treated as equivalent. And finding the relationship between those data and how they can be used together is something that ARGA has been concentrating on as well. And so out of that, we derived a number of aims that were based on the community and also understanding the data that we were working with. And when we were talking to people, we found that what holds them back from using data or interoperating with it, even once they've found it, is that they have a reluctance perhaps sometimes to trust data. And the other aim that we were trying to solve when we were developing ARGA was to make sure that people could have feelings of trust and that the trust would be enhanced by understanding the quality of the data, whether or not it came from taxa and that those taxa were well identified, and also that there was sufficient metadata for people to actually make decisions about whether or not they wanted to use and reuse the data that they'd found. So out of that, we came up with a number of different strategies. I won't go through them all here, but I just wanted to reassure you that we have actually gone through quite a long ideation process with this. And one thing that we did discover was that we were going to have to come up with a bit of a new model for how we were approaching building this index. As you saw from the previous slide, the data are nested, and those data fit within what are essentially nested populations from individuals that fit within a population which fits within a species, which again fits within a nested structure of a taxonomic hierarchy. And I should say from the outset, sorry, but I'm actually a morphologist and a taxonomist, and so taxonomy gets me a little bit thrilled to think that I might be able to classify something and put it into hierarchical structure. So this is born out of that need for not only a hierarchy for the animals or the plants that we're dealing with, but also for the data itself. We felt it was really important that our model did reflect the hierarchical nature of the data, and it was also able to integrate data from a number of different sources. And they were all actually different. So data from NCBI is not the same as data from BPA. It uses different field names. It's not necessarily something that you can just mash up against one another quickly and easily. We also wanted it to be able to interoperate with biodiversity data. And those data are stored within ALA, where I'm from, in what's known as Darwin Core format, and that is not a format that genomics data are generally stored in. And so our big challenge was to try and align all of these things together. So this is just part of the model process that we went through, and what we've done is we've adopted an event-based strategy, and the way I sort of like to think about that is that something exists in time and space, I exist in time and space. I'm here right now. But I also existed in time and space when I was at university, and you all did too. And a number of things have happened to us throughout our life journeys, and it's exactly the same with the sort of data that we're dealing with. It's coming from specimens. They were once in the field, they've been collected, they've been subsampled. Different things have happened to them, different treatments have happened in the lab. And at each of those points in time, people have interacted with it, and there's a whole heap of metadata around that. And for us, making sure that we track this provenance chain is what is crucial to us building trust in the data for users at the end of the day. So this is just one of the models, we won't worry about it too much. But essentially, as I said, the aims that we were trying to address when we were building ARGA was to help people resolve the taxonomic uncertainty that they were dealing with when they were finding data from places like NCBI. And we did that by incorporating authoritative diagnoses and taxonomic structures from things like the Atlas of Living Australia, but also the National Species List and the Australian Faunal Directory, and using them as our primary sources. We've also included informal taxa, like SPA or manuscript names, because these are generally not included in things like the AFD, but an enormous amount of genomics data around them is there for that reason. We also wanted to include some tools to help users with their visualisations of the data before they interacted with it, so that they could decide if they wanted to go through the effort of downloading it. And to that end, we've included the ability to look at trace data and things like that. And we've also been trying to increase the amount of metadata that people are able to discover by linking up with occurrence records that are from the ALA and from museums and herbaria. And so with that in mind, I would invite you all to go and check out app.arga.org.au, where you can have a bit of a play with it. I will tell you from the outset that there is not much other than frogs done to a high level of data confidence in there, but we're building up the data set as we go ahead of our launch in September. And I also wanted you to have a go when you go to ARGA at 3 different use cases, which I'll very briefly tell you about. The first one is that you can look for a taxon, and you may want to look for it by common name or scientific name or something like that. In this example, I'm looking at cane toads, and I will very briefly just show you that it works. And you can get all sorts of information and have a look at it. The other thing is that you can also use the icons to look at things by traits, and in this example I'm looking at frogs, and we've used taxonomy as a trait there. The other thing that we are able to do is look at coverage within geographic areas, and this is something we'll be working up in the next iteration. But you'll be able to see baseline data for how much genomics data is available for any particular taxon in any particular geographic area. So in summary, ARGA's actually trying to solve a very complex problem, and that is to align all sorts of different data, their biodiversity data, their geographic data, their traits data, as well as their genomics data, which is different no matter where it's coming from. And with that in mind, I would ask you to go and test the app, and it's available at app.arga.org.au. And if you would like to talk to me, I'm Kathryn, and you can reach me at kathryn.hall@ciro.au. And also here with me today is Kiva Connolly, our scientific business analyst, and we'd love to talk to you more. Thank you.
Good morning, everyone. My name's Tom Bruce, and I'm here to represent the Wildlife Observatory of Australia. And I'm going to talk to you today a bit about what that is, what we do, and how we can all get together to achieve it. So I'm here to introduce TERN's newest observatory, the Wildlife Observatory of Australia. And what we are is a collective of scientists, and our aim is to monitor Australia's vertebrate populations primarily using camera trap data. I'm going to talk through why it's needed, what we're doing, and how we are going about achieving it. So as I'm sure you're all aware, Australia is currently undergoing a wildlife extinction crisis, predominantly in mammals. In the last, since colonisation, there's been around 38 mammals go extinct in Australia. An example of that is to the right, the extinct Tasmanian tiger. And now there are a variety of causes of these extinctions, and they're mainly split into long and short-term drivers. These include climate change, habitat fragmentation, and short-term like floods and fires. Now, while it's important to monitor mammals whilst they're going extinct, other reasons to monitor wildlife include assessing the impact of development. So before any work is carried out, an environmental impact assessment is needed to understand what the impact on wildlife could be of the development of infrastructure like roads and mines. Now, while these two aims might look disparate, they have a commonality. They both need baseline data and resampling to track the change of wildlife populations through time and understand the impact of mammalian extinction, but also what developing a road might look like on a mammal community or wildlife community after it's been completed. Now, to gather this data, historically, we would have turned to spotlights and tracks and signs. There are several drawbacks with this, including it takes a really long time and a lot of money to gather the data using people to directly look at wildlife or look for their tracks and signs such as dung and nests. It particularly isn't very good for cryptic species like feral cats, which are going to avoid your spotlight, and therefore we need to turn to a new method. This means that the use of camera traps has dramatically increased through time, and camera traps are great. They provide longer and cheaper surveys. It's like tying a volunteer to a tree 24 hours a day to look for your target species. They're particularly good for cryptic, diurnal, and nocturnal species, as they don't really specify what they're looking at. They just look for a change in movement and temperature in the environment, so you can survey the whole community effectively using one method. However, this comes with a small asterisk that one survey can generate over 1 million images quite easily and quickly. And this is the big downfall of camera trapping. Images are really easy to get hold of, but what they are hard to do with is to actually turn them into a usable data set to generate great inferences from your data set. So this is where WildObs was born. About a year ago today, we gathered together 35 participants from academia, non-government organizations, government organizations, and we came together and decided that we need to form an observatory, and together we're going to move forward, and I'm now going to talk with you a bit about what that actually means. So to get a little bit of background of the state of camera trapping within Australia, we had to look somewhere, and we started at the ALA. So we actually scraped the ALA database for records of terrestrial vertebrates that contained the term camera anywhere within the record. And what we can see with this figure here is that the grey hexagons represent we didn't find any records of camera traps in a 500 kilometre squared area. The blue is less than 5 records, and the pink goes from 6 up to 230 in the red. And what this is telling you is that even in New South Wales, which has the most records with 3,120, there's a very clear bias. We're biased to the coast, and we're biased to mainly around urban areas. But what's really important here is there are clearly gaps in monitoring across the country, and so this represents an opportunity to fill these gaps. But importantly, there's lots of cameras, but they're not connected into a network. They're all disparate entities potentially researching the same question, but drawing different conclusions because they're not leveraging the power of one another. So we view the WildObs pipeline basically as 3 key steps, which I'm going to break down a little bit more, and that is we're going to take images, convert them into a standardized database that's findable, accessible, interoperable, and I've forgotten the R. And we're going to then bring state-of-the-art analysis to bear and help people get the most out of their data. So the first step in this is establishing field sampling standards so that everyone's gathering data in the most standardized way possible. The second step is then to actually leverage AI and machine learning to help us attribute which species are on our images and take some of the burden away from scientists, and then use human oversight to verify that. The third step is then to collate all of this data together and keep it in one standardized place, and we're locating that database at TERN. And the final step is to take advantage of advanced analytics such as hierarchical and occupancy models so that end users can then either generate great data for a peer-reviewed publication or just have the most robust metrics to assess adaptive management strategies such as feral pig management. So I'm going to take you through each of these steps now. The first of which is standardizing and securing images. So many camera trap surveys fall down at the deployment phase, i.e. you don't know what you're doing and you've sort of gone out to try and look for something. So we are developing a series of manuals to help people identify their research questions and how best to deploy cameras. We're currently proposing that putting a camera on the road to target predators and species that use linear infrastructure is good, and then you'll have your second camera in the habitat to potentially target species that are going to avoid linear infrastructure, potentially due to a fear of predation or encountering people. We're also developing apps to store standardized metadata. As other speakers have alluded to, metadata is really important to allow the datasets to talk to one another, and people gather it in a range of different ways. If we force people to standardize where cameras were placed and how they were placed, we can then get datasets to talk to each other more easily and more readily. The next step is backing all of the images from the cameras and also the metadata into the cloud. So many images are lost every year because of hard drive failure and people keeping them in places but not backing it up. And also metadata gets lost in field notebooks all the time. So forcing everything into the cloud keeps it much easier and straightforward for everyone. Now, anyone that's ever touched a camera trap and tried to survey with it will tell you that processing images from pictures to a usable data set is hard and a massive time sink. We're proposing to speed up that process via 2 steps. The first is using the help of AI and machine learning with our partners Environmental AI and Wildlife Insights to actually remove the blank images and keep only images with wildlife in it. The AI models can then propose a potential ID of what they think they've got, and these will then be passed to the citizen scientists, thanks to our friends at the Australian Museum, to then verify what the machine learning algorithm is labeling the image as, and once verified, these can then feed back and improve the performance of AI. This then means that you've now got an image that works and translated into a database in record time and is now ready for analysis and use further down the pipeline. Now, once we've got all of our lovely images from our stored camera traps into a standardized format, we then want to host a data set at TERN which represents all of Australia. What this means in reality is that then participants from academia, NGOs, local government and state government can all feed their data to WildObs at TERN, and then in return they will receive standardized data sets, custom analyses and robust feedback. But what this actually means in terms of a great benefit for everyone is that the federal government, the Atlas of Living Australia and the Threatened Species Index no longer have to go and ask each individual party for their data. They can just come to WildObs at TERN to receive this data in a way faster time than ever before. In terms of public engagement, we plan to use a Threatened Species Index style website for overviews, so anyone could come and look and see where the sampling in Australia has taken place, have a look at some trajectories for their favourite species, along with other overview statistics. Now the next bit, and that's really unique, is that we're proposing a portal where approved users could come and pitch a species project. So they wanted to look at quoll trajectories in Australia. They could write to the Wildlife Observatory. This would then be passed to any data users that have records of quolls, and then the person that owns the data then approves or denies the access request. But what this means is it's taking out the step of having to go to individual institutions to look for people. You can just come to one place and pitch your species project and leverage other people's data. Now I've sort of gone through the nuts and bolts of what could be possible using a database. I'm just going to encourage us to move into the future and think about what could be possible. What we're proposing is something similar to Snapshot USA, where we could create a database and a network of researchers. What this figure shows is the detections of mammals across the entire United States of America. Now imagine if this was possible in Australia. You could now analyse data at a continental scale to understand, for example, how populations of feral cats are performing across the entire country on an annual basis. You could also use it to look at endangered species such as quolls and see how they're responding to management interactions at a continental scale. What we think is really important here is that we're facilitating collaborations between industry, NGOs, government organisations and academics that have previously been really difficult to get people to talk to each other because there's just so many people scattered all over the place. Now I'm just going to run you through a quick case study to show that the Wildlife Observatory is open, ready for business, and we're here to help people, and that's the case of the WWF and Google Eyes on Recovery programme. For those of you that don't know, they deployed cameras to monitor the wildlife response to the 2019 Black Summer bushfires. What this means is they deployed 1,023 camera sites across 10 landscapes, which accumulated 240,000 trap nights of effort, resulting in 7.5 million images to be processed, which is about half a million independent captures on these cameras based on this data set. Now, where we came in was helping them actually manage and analyse the largest camera trap study in Australia to date. What this graph on the right is showing you is the diamonds are individual camera trap sites, and you can see it's a really great coverage across the eastern seaboard of Australia and Kangaroo Island in South Australia, with the GBAM layer underneath showing you green areas that didn't burn and red areas that did burn. Now, the important point here is that once WWF provided the spreadsheets of data, we were able to analyse that data set in 8 weeks, which is probably one of the fastest times ever for such a big data set to be turned around in time for the ESA conference in 2023. What this actually means is that we modelled and analysed data for 93 different species and provided the overview statistics for their camera trap survey. So what's the future of WildObs? Well, our home is at TERN, and I want to thank all of our collaborators from TERN for their continued support throughout this project. But ultimately, we're ready for more data. We can do it, we've demonstrated we can do it, and we've carried out help for WWF, so we can receive data already and start processing it for everyone else. And our overall aim is to create the largest network of camera trappers in Australia to enable continental level analysis. To put our money where our mouth is, we've applied for $2 million in ARC research grants through the LEAF funding scheme, which means we're already connecting with NGOs, government, academia, and other stakeholders. And please come and talk to us. Myself, Matthew Luskin, and Zachary Amir are all here in the crowd today, so we're ready to hear from people, hear your ideas, and start building collaborations to move forward. I'd like to say a big thank you to all of our collaborators, particularly the universities on the slide, who have really come forward and helped with the ARC LEAF grant funding, and welcome any questions from the audience. Thank you.
G'day everyone from Curtin University in Perth. Unfortunately I couldn't be there today. I've got a lecture on. I couldn't talk live or attend and had to do this pre-recording. But Mika Vanderheide, who did the bulk of this work, will be online and will be available for questions. So first of all, I'd like to acknowledge the Whadjuk Noongar people whose Budja this work was conducted. Offer respects to the people and their elders past and present. Mine site monitoring, why are we interested? It's a big business. To give you some numbers to put this into perspective, the footprint of mining in China alone is 3.7 million hectares, and that's around about the size of the Netherlands. So the scale and cost of restoration needs to make sure we are getting bang for our buck, and to do that we need to monitor. So there's three main reasons why we monitor. Firstly, to identify whether restorations are working or do they need to be modified or changed in some way. Secondly, to answer questions about new treatments or the return of organisms or processes to a site following restoration. And thirdly, and increasingly importantly, to provide evidence to stakeholders and government regulators that goals specified in environmental impact assessments are being achieved. So how do we determine when restoration efforts at a site are on the right trajectory? And this is a key question, and have reached a suitable threshold for a regulatory body to sign off on. So common goals set for mine sites following closure include restoration of structure, diversity and function of the disturbed ecosystem. Plants are very much the focus of this monitoring, and soil microbial communities and fauna are often neglected, and their restoration or recovery following the cessation of mining and restoration is really poorly understood. So we've argued for a number of years now that a more holistic model of restoration monitoring is overdue, well and truly needed, and such a model would enable assessment of everything from soil microbial communities to charismatic mammals. So some of the work my group has been conducting over the last 5 or 6 years includes just looking at what substrates we can use to detect biodiversity at a site. How do we actually sample those substrates? We've used everything from paint rollers from Bunnings that have been adapted for what we want to do to just swishing around filter papers in water. And then we've applied these learnings to monitoring restoration trajectories. So based on all this work, we have developed a suite of 5 points that you need to consider when designing eDNA-based surveys of restoration, and published a review on this work, or a policy paper. First of these is substrate selection. It's really critical that you understand what species you're likely to detect in your particular substrates. Here we looked at scat, pitfall traps, vein traps, plant material and soil, and you can see that some substrates are better at detecting certain taxa or more taxa than others, and that combination of substrates is probably going to enable the detection of the most taxa. We've also looked at novel substrates, a recent paper where we looked at flowers and we're able to detect from flowers on a variety of taxa, including birds, invertebrates, and mammals. Sampling design is another critical area when looking at using eDNA to monitor restoration. Mine sites are really designed with monitoring in mind, and reference sites may be distant from the area being restored, or you may have issues with spatial autocorrelation given the location of reference sites. We also found from our review that monitoring is typically conducted at one time point, not through multiple times of the year, and often in studies when monitoring was conducted is not even mentioned. Thirdly, assay selection is absolutely critical. You can use assays that target specific species or groups of taxa, or you can go more broadly, for example, using a vertebrate-specific assay. And from our recent study on the flowers that I just mentioned, we compared 12SV5, which is the standard vertebrate assay, to one that we developed that targets bush birds, and found that the bird assay that we developed outperformed the 12SV5 assay, and highlights the issues of not understanding what taxa your assay can amplify, and the potential for false negatives. Fourthly, data interpretation, and this is something we really want to get into now. How do you determine restoration success? Where do you draw the line in saying whether restoration is successful or stalled? And this is really challenging, and as I said, something we really want to focus on with our work now. Finally, reporting standards. It always astonishes me how poor the mining sector is at recording how they've conducted monitoring and their storage of data, etc., etc. This is really crucial with eDNA-based monitoring, as the way you collect the samples or store the samples or analyze the data can really change the findings of your study. So with eDNA, you really need to understand both its strengths and weaknesses, need to manage expectations. A lot of the resources sector see this as a silver bullet, and perhaps it's not. eDNA is best in some situations. Long-standing approaches may be most appropriate in others, and often it's a combination of both traditional and molecular methods that are best to measure biodiversity. So have we got the tricoder yet to identify species? No, but recent developments, including AirDNA, means that we can sample remotely over long time periods, and then using more high throughput molecular methods, we can generate data quite quickly. I'd just like to thank mainly Mika Vanderheid, whose work this is largely based on, but also the other students who were involved, and also the cast of thousands from both Curtin and other universities, and also funders. Some email addresses there. Please email if you have any questions. Mika will be online now to answer your questions as well. Thank you. Bye.
Hello everyone, this is Zefang Shen from Soil and Landscape Science Group at Curtin University. I would like to introduce our study on miniaturized spectrometers for assessing soil health indicators to support mine site rehabilitation. Mining can cause several disturbances to soil, and post-mining rehabilitation requires monitoring a wide range of soil physical, chemical, and biological properties as soil health indicators. Visible and near-infrared soil spectroscopy is rapid, accurate, and cost-effective, and it could be a solution for cost-efficient monitoring of soil health indicators. And also the spectrometers in this range are becoming smaller and cheaper. However, their performance to predict a wide range of soil properties has not been assessed. Therefore, our aims are to compare 4 miniaturized visible and NIR spectrometers with a benchmark spectrometer to estimate a wide range of soil chemical, physical, and biological properties as soil health indicators. And to this end, we assessed the modeling with different algorithms, and we assessed the accuracy of the modeling with the spectra from different spectrometers. We also assessed the repeatability of the spectroscopy modeling when it is done with spectral replicates. And in the end, we derived an overall performance score based on the accuracy and the repeatability. We sampled soil from 7 mining sites in Western Australia, and this gave us samples from 56 plots. And in each plot, there are 5 sub-samples, so this gives us totally 280 subplots. We measured soil physical property, biological property, and chemical property. In total, there are 29 property measured. We used 1 visible range miniaturized spectrometer. This one here indicates the range of the spectrometer. We have 3 NIR spectrometers, and this one, and this one. And we also show the soil spectra collected with those spectrometers. We compare this miniaturized spectrometer with the benchmark spectrometer, which covers the full visible and the NIR range from 350 to 2500. Because the spectra from the different ranges can carry different information to make the predictions. So we also considered combining the visible spectrometer with the NIR range spectrometers, and here I've combined the spectra. To assess the accuracy of the spectroscopy modeling, we developed models on the 56 samples with average spectra. We modeled 29 soil properties using 7 machine learning algorithms with 10-fold cross-validation. And then we assess the accuracy using only the best performing algorithms out of these 7. To assess the repeatability, we did the modeling with the spectral replicates on those 56 samples, and we modeled the 29 soil properties using the best algorithms from here, and also we used 10-fold cross-validation as well. The repeatability is assessed based on the accuracy difference between the modeling with those spectral replicates. In the end, we derived an overall performance based on the accuracy and the repeatability. To assess the modeling, we use this concordance correlation coefficient score. It ranged from -1 to 1, with 1 denoting perfect agreement. And we also divided the accuracy into different levels to help with the interpretation. Yes, here we compare the different algorithms. We have 7 algorithms here. It's quite clear that these 4 algorithms, performed quite well for the physical and the biological properties, and they are clearly better than the rest 3. So these algorithms are the partially least square regression, QBist, Gaussian process with its linear and polynomial kernels. Similarly, for the chemical properties, we could also see that these 4 algorithms were much better than the rest 3. So we are going to use these 4 algorithms to assess the accuracy of the spectrometers and the repeatability of the spectrometers. Here we show the prediction accuracy for the 29 properties with different spectrometer and combinations compared with the benchmark spectrometer. You'll see, as I mentioned, it's from length 1 to 1, and higher you'll see means better accuracy. We could see for the visible range spectrometer, it predicts the sand, this point here, silt and clay pretty well. And for the NIR spectrometer, actually it predicts most of the physical property and some of the biological property, and most of the chemical property quite well. And also, if we combine this visible range spectrometer with those other NR range spectrometers, we could see the prediction has been improved in many cases. And here, some chemical properties here as well. So in total, these miniaturized spectrometers and their combinations was able to predict 24 out of 29 soil properties with moderate or greater accuracy. Here we use this delta ROSI to assess the repeatability of the spectrometers. This is the accuracy difference between the modeling with two spectral replicas. A smaller delta ROSI means better repeatability. And this benchmark spectrometer was the most repeatable for almost all the properties, and the visible range spectrometer was quite repeatable for the physical properties, but didn't perform well on the chemical and biological properties. The NIR spectrometer was quite repeatable and similar to the benchmark spectrometers. But however, when they are combined with the visible range spectrometer, and in some cases the repeatability was worse. In most of these cases, these properties could not be predicted well. Based on the assessment of the accuracy and the repeatability, here we derive an overall performance score. This first item is the mean accuracy across all the properties. And here, this one is for the stability of this accuracy. And this is like the mean repeatability across all the soil properties. And here is the stability of this repeatability. Yeah, if we look at the overall performance score, of course, the benchmark spectrometer was the best, but the spectrometer with wider ranges, they were kind of similar to the benchmark spectrometer, and also the combined spectrometer will also have similar scores to this one. Yeah, to sum up, in this study, we compared miniaturized visible and near-infrared spectrometers and some of their combinations with the benchmark spectrometer in terms of the accuracy and the repeatability of the spectroscopy modeling for soil health, for predicting a wide range of soil health indicators. So the conclusions are these miniaturized near-infrared spectrometers and their combinations with visible range spectrometer performed similarly to the benchmark spectrometer in terms of the accuracy and the repeatability. And those miniaturized spectrometers was able to estimate 24 out of 29 soil properties with moderate and greater accuracy. Therefore, we believe miniaturized soil spectroscopy in the visible and near-infrared range could help collect soil data at finer and at finer spatial and temporal resolutions to support soil health diagnosis and to support mine site soil rehabilitation. Thank you so much for your attention. I'm happy to answer any questions. Hello, everyone. Good
morning, everyone. So my name's Sommer. I'm a software engineer in the environmental informatics group at CSIRO. I'm based here in Brisbane at the at the Dutton Park office. So again, I'd like to begin by acknowledging the Turrbal and Jagera people, who are the traditional owners of the land that we're meeting on today, and pay my respect to their elders past and present. So this is the problem that we're going to be talking about today. Not surprisingly, very high resolution time series raster and gridded datasets with a, sometimes with a high temporal resolution as well can get can have a very large size on disk. For example, the Somerset actual evapotranspiration dataset has a size of over 4.7 terabytes on disk. And if you're a scientist, a researcher, analyst, if you want to actually use this, use our published data for something practical, it's a bit impractical to download the whole data set if you want to just look at, a subset. So what I'm talking about there is if you want to, if you're interested in a single point to drill a single pixel over time, or if you're calculating zonal statistics using custom polygons, or if you're, want a bounding box over time to create a smaller data cube of a region of interest. So we've been thinking about what we can do about that in TERN Landscapes. Some of our users have expressed their desire to use our data products in a more targeted manner. So their common requests are, as I mentioned before, the ability to subset data spatially using provided polygon bounds, or aggregate statistical data over time given custom polygons, or extracting a time series from a given point. But it's not practical for us to have pre-computed, stored representations of all of the different permutations that people could ask for. So ideally, these requests should be dynamic, with the expected data generated and delivered on demand. You're probably thinking, isn't this a solved problem? Don't we have a solution to this? Yes, well, OGC to the rescue again. Of course, they have a web specification for this. This is the OGC WPS, or Web Processing Service Specification, which was originally published in 2007. So this specification describes a system where a service can advertise a collection of available operations or processes that can be parameterized with given inputs and outputs, and the server can process that request and respond when it's complete, giving the operation results, and optionally a payload, a data payload to download. So this is our implementation of that specification. Our implementation is called FuncWPS because it's the TERN Landscapes functional WPS server. We didn't invent this from scratch. It's implemented in Python on top of the PyWPS application framework. Just to be clear, this service, it doesn't have its own user interface. It's a headless service with only a machine interface API that you can interact with it. The application itself is hosted on a cloud virtual machine in our allocation within the Nectar Research Cloud. For best performance, we've found that it's important to have this application hosted physically as close as possible to where the data is. So in our case, the data is stored in the Nectar object store. So we've found that if we host the application in the Melbourne region of Nectar, that gives us the best performance. So this is a bit of a breakdown of how the WPS server actually processes the requests. Firstly, it tries to identify what the requested operation is. It then goes and pulls in from our object store all of the data that it needs to use, loads that into memory, and then to increase performance, it can use multi-threading and multi-processing processes to speed up the computation, and then it can generate an output, whichever process that you're doing has different multiple output formats that you can choose from, and then it responds back to the caller saying this is done, and then you can serve up the file. All right, so a cool system. How can we use this? The delivery of this TERN Landscapes WPS function server, it's useful, as described, for people who can know how to craft their own, if you're familiar with the WPS specification, you can craft your own WPS requests and send them into the process queue, pull back the response, unpack the response, analyze it, or if you have specialist GIS software that kind of has that general arbitrary WPS request functionality, but we've found it's mostly not very useful for the vast majority of users. But to make it a bit easier, we have published some sample Jupyter notebooks. So they're Python-based examples on GitHub, where it shows worked examples of how you can set up your inputs, your polygons, submit requests, pull back the responses, extract and visualize your results. But what about the large majority, the large body of users who don't want to try to use the programmatic access methods? Is there a easier or more accessible way? Yes. So shown here is our Landscapes Data Explorer. So this is a Terrier.js-based web app that we released about 12 months ago. It allows you to visualize and explore TERN landscapes datasets. And recently we've tapped into the built-in WPS feature in Terrier.js to allow us to send requests and get back responses through Terrier.js. You can drag on your own points and polygons, and it sends the requests dynamically. But what if you don't want to use this full-featured application? It's a heavy app to run in your browser, and you probably, if you don't need all the visualization capabilities. Finally, we do have an even easier way. So this is the TERN Landscapes cookie cutter service. So this acts as our kind of missing user interface to the WPS server. So you can drag your shapefiles onto the drop zone, choose your target data set, choose your date range, hit send, and it does it all for you in the background to make it as easy as possible to access these data sets. All done?
Okay. And now from the very complex to something much simpler and about you, I think, the actual researcher. So, very pleased today to be able to talk about some tools that I and a rather brilliant research team have been working on developing to enable researchers and research teams to better apply and engage in the open science practices that we're all supposed to do. But first, I'd like to acknowledge the traditional owners and custodians of the land and sea in all nations. We honour their profound connections to land, water, biodiversity and culture, and pay our respects to their elders past, present and emerging. And in my case, it is standing here with to acknowledge the Turrbal and Jagera people. Now, I will mention here that FAIR and CARE are two acronyms that absolutely go around everything that I'm going to talk about in the future. And if you don't know what they are, I'll talk to me later, but many other people do. Okay, so back in 2021, the UNESCO made some open science recommendations, namely that increased openness will lead to increased transparency and trust in scientific information. We've heard about trust already this session. Very important for the aims of TERN and such like, as well as for us. Now, the key pillars of Open science, as they continued to talk about, were to enable open scientific knowledge through, of course, our traditional scientific publications, but increasingly research data attached to those publications, software and source code, hardware, and hence open science infrastructures, science communication, indeed multilingual, and open engagement of societal actors, the citizen scientists we've already heard of, and open dialogue with other knowledge systems, which we've also heard about this session. Okay, but from the very beginning of the research process, the researcher both contributes to open science and takes advantage of the open science practices of other members of the research community. It's a win-win situation, or hopefully it will be, and so UNESCO would argue, and I think we might well agree. And keep an eye on that snail, because I'll come back to that later. Okay, so we are talking from a project that we got money for, looking at from the Belmont Forum, which is an international intergovernmental group which makes periodic decisions about collaborative research actions, and in our case, it was on science-driven e-infrastructure innovation. And we have funding from Brazil, France, Japan, and the United States, with collaborators from Australia, and support, of course, from a synthesis centre in France. And we're looking at building new tools for data sharing and reuse through a transnational investigation of the socioeconomic impacts of protected areas. Some of you have already been approached by me with respect to this, gathering more data. So, didn't press hard enough. What did science-driven e-infrastructures innovation mean for our project? So we created two strands. One was a synthesis science strand, which would go about the business of using existing data, hence the title, and another would be the data strand. And these two strands would be over 3 to 4 years, eventually, due to COVID, maybe 5, would learn from each other. And the data science team members were to ensure a comprehensive data and digital output management plan was employed. Now, this was mandated by the funder, the Belmont Forum, and of course, therefore, each of the science councils in each country, and would develop new tools for data sharing and reuse. Now, the synthesis team, just to give you the picture and the context in which we were working, was a team in these mainly 5 countries, but we also had a project in East Africa, who were looking at developing algorithms to predict socioeconomic indicators around the creation of the impact, a Barchi process, where a before and after impact, the creation of the protected area, was monitored via census data, the ground truthing, satellite images, and of course the algorithms were produced. And this was the great idea via deep learning and AI. And that has been progressing very nicely. Of course, the research data workflow, and you've seen one from Bronwyn this morning. This is one of the other early published ones by Bill Missioner of Data One, who produced this kind of nice little plan of how the data workflow works through a project. And this was what the data science team was to talk to the research team and understand how, in this particular situation, this exercise was performed. However, thanks to some work done here in TERN that we published back in 2015, when you're dealing with a synthesis project, you skip a lot of that planning and hypothesis setting in the same sort of way as you do when you're going to collect your data, raw data. And so back in 2015, Guru and I looked at, and some others in the team decided that really there were 4 or 5 steps that we needed to be concerned about, and we have found that to be the case in the Parsec project. namely, and you can see in the middle there the sort of composition of the team that we're dealing with. And of course, if you look at that as multinational, we've got a lot of cultural differences which have become very interesting over time, have had to be negotiated on top of the interdisciplinary, transdisciplinary career stage and all those sorts of normal things. And so we have this step, which is the ingestion of data, the identification, ingestion of and acquisition of data, and making that easy is terribly important. Trusting that data, of course, collating and aggregating it. Hopefully the metadata will help in that selection, and we had to find temporary storage for that so that we could do the analysis and synthesis, or at least the synthesis strand could do it, and then visualise that for our various actors, as well as publish in the traditional way. But then, of course, we also have to have a data repository. Now, of course, this forward flow, both expressed by Mishner and in our particular diagram, is notional. There's lots of toing and froing and corrections and throwing out. So that is actually quite important, because we do want to record our failures, or at least we did. And we continue to. So the first step that we were asked to do, as I mentioned, was to develop a data and digital output management plan. And how do you do that? Now, one of the things that and the Belmont Forum via a paper by Wade Bishop, which my apologies, I didn't put the link there at the bottom, has produced a paper saying this is what it should be. We appear to be the first people to have really tested that in a practical sense. And one of the things I didn't expect, I must say, in the DMP, as we used to know it, is the establishment at the start of how you're going to talk to one another, your team communication, what you're going to use. So this requires a dialogue with your team members. And then, of course, one of the things that's absolutely fundamental is to have your individual orchids. Remember, we're engaging in open science, your individual orchids and your institutional RORs ready and recorded immediately, so that you can keep on doing that for any conference paper, whatever outcome you may be doing. So that was number one. Number two was getting the data, of course, and collating and harmonising it. We ended up using Google Drive as our primary temporary storage to do that. And that's become a rather large and repeatedly needs a AAA read me, where will you find things sort of thing. But we can use it across all countries. And that was very important. But of course we had to have proper temporary storage, shall we say. And for that we looked at the Open Science Framework and the Amazon Web Services. I did try flying a few others, but you need acceptance that these tools are okay. So when you're building the data management plan at the beginning, you've got to have a conversation with your team members, because they've got to be able to use it and be willing to use it. In the end, we didn't use AWS very much at all. Neither did we use Slack, despite everyone's initial enthusiasm. So it's interesting how the best laid plans get revised. With respect to analysis and synthesis, we chose GitHub, and we have Parsec World as our GitHub handle and for our software development. And that is, of course, publishable via Zenodo. So we get a cross-reference between the two. And every conference paper, for example, this one might go into our Zenodo Parsec community location. So we get a DOI for every interim presentation and output, but we also have, of course, publications in the formal sense going into normal journals, which are linked through Crossref and which link back to our ORCID, which is very important for many of our participants. Not all, but many of them. Okay, and then, of course, we're going to produce the data, we're using the data, we're producing the data, and we adopted CC by 4. Remember, we had the US people who, have CC 0, but CC by 4 was a general international standard that was acceptable. And of course, we've got the software licensing codes there. that we chose. Now, all of this had to be done at the beginning, including the establishment of a repository, and that is actually really important. I've been rather having fun going to the Research Data Alliance meetings and learning lots of things about this sort of process along the way. And one of the things is to have a Core Trust Seal accredited repository, and I believe TERN, if not already, but is almost about to get such accreditation. And the EDI, the Environmental Data Initiative, funded by the NSF, coming out of Santa Barbara as it happens, has got that accreditation. Now, the point of that is that you have a partner. So if you actually start at the beginning of your project, be it as an individual or research team, but particularly for a research team, and you've identified a repository, you start having a dialogue with the agent in your repository, the person in your repository, because one of the common problems that repositories complain about is someone's published a paper, got it accepted, oh heck, before it will be actually published, actually open, I have to deposit the data. Panic, panic. And they find the data's in the wrong format, you've got to hold the journal up, on and on it goes. Whereas if you've actually started at the beginning with your metadata, with your data organisation in a way that will make it fair and usable and interoperable by in the repository sense, you'll be much better off. So having an early discussion with the repository is one of the neat things that DMP or DDOMP will give you. And we've got an example of, I've put there an example of that work of our workbook filled in on Zenodo. The diagram, by the way, comes through a paper that we're currently revising. Now, the difficulty is how many pages in that DMP? 19. Most of the researchers, of course, in our team, we're learning from each other, said, this is crazy. We can't do this. You've got to have a delegated person, but we're not going to pay any attention. Okay, so this is where we started to break down the DMP for the researcher. And there's the snail again. We're looking Fibonacci series, if you like to think about. What we found, what we're going to do is start at the beginning with the person, the researcher. So engaging with open science, getting themselves known, what matters to them, what is the core business. Then you step forward to the research team or lab and you move further to the community and beyond the community. So what we've been developing are checklists for the first 2 phases so far. And so they're out and published and you can have a look. So useful checklists for the researcher, there are 3 of them. I'll show you just one. And this is optimising your digital presence. You in the open science world, if you're an early career researcher, this is what you should be doing. If you're really late career, you probably don't care. But for me, it's, I really was worth something at the end. So we've got these sorts of checklists downloadable. We may well have little videos. And of course, then we have them for the research team as well. So this was actually the most complicated of them, was how to practice open science and have a code for your team. So where does that leave us towards the end, Nevill? We've learned from researchers about what they need. Remember that pussycat? We got that a lot. The researchers have gradually come along with us. By the way, I bridge both programs. Simplicity, ease of reference, no steep learning curve, that sort of thing. You've heard that before. We've exposed the development of the DDOMP tool. We've got a paper in the press. And we've developed these checklists, which are in their first view to the team and the wider community. Each checklist, remember, one of the aims was multilinguality, has been translated into 4 languages. Now, to be careful, we could send it into a Google Translate thing and get hundreds of languages, but actually the refinement of getting the words right is quite important. So 4 is pretty jolly good. And of course, Australian American are another 2. And the DDOMP in each checklist is openly available through Zenodo, Parsec Community, as models and guidance for others to engage in hopefully easy open science practice. And I've put the links to our material here. I would suggest you go first most easily to the AGU website, American Geophysical Union website, as it is rather pretty, as well as all aggregated in one place. So thank you very much, and thank you to our funders and partners.
I'd like to invite all our speakers back up to stage. We've got some questions. So a question for Kathryn or Tom. What do you think are the key roles and skills needed to build a successful team to deliver the Atlas or a service like Wild Oz? Do the microphones work? Yeah, perfect.
Wait for it to warm up. Okay. Look, I think one of the most important things is that you understand that you're stronger. I mean, it's a cliche, right? But you're stronger together than you are each individually. And what has been the best thing on the ARGA team is that we've got people from all different backgrounds, and I think it's one of our strengths. You know, we've had people from strong scientific backgrounds, but also people who've come from a more commercial space. And we've been very open on our team. I have to say, we're a happy team, which is lovely. But we know that we're all different, and we take the time to listen to the perspectives that each has and the strengths that they can bring. One of the key things that's been really good for the ARGA project is that we have engaged early on with not only scientific business analysis, but also having a UX, UI designer as well. So that's user interface, user experience designer. And I think that's been really important for us, just to help us be always conscious of the needs of other people, because I might think something's lovely, but you know, I'm not everyone else. So.
Sure, yeah. I guess I can add a little bit to that, but I sort of echo the same sentiments. But the biggest thing for us was making sure that the type of analysis or materials that we're going to develop is actually what people want. So doing a summit, bringing everyone together and saying, hey, you all come and represent a diverse range of backgrounds from government, academia, NGOs. What is actually useful to you? Because there's no point building something that people aren't going to use and isn't useful to them. So I think, yeah, developing your team with that in mind and making sure that everyone knows the common goal you're pushing towards is really important.
Do we have the online participants as well to answer questions? Okay, well, that will make it easier. So Ashley, what about the interoperability of the layers with Google Earth Engine? Can you speak to that?
So with Google Earth Engine, my understanding is, so one of our data products, the Somerset actual evapotranspiration data set, the kind of really big one that I was talking about in the presentation, that one is actually generated on Google Earth Engine, and its primary presence is on Google Earth Engine, so it is available already as one of their, I forget what they call it, one of their open, like Google Open Exchange data sets that you can use. We don't have any plans of putting our other TERN Landscapes data sets up into Google Earth Engine. Of course, if people approach us and ask us about it, it's something we could think about it.
Thank you. And a question from Belinda for Ashley. What's the plans for expansion and adding more data sets?
Adding more data sets to the TERN Landscapes Explorer. So at the moment, I think there's 3 data sets up there. So you've got this Somerset actual evapotranspiration data set. There's the SMIP soil moisture data set, and the Soil Landscape Grid of Australia soil classifications. So they're kind of what we see as the 3 kind of flagship TERN Landscapes products. There are quite a few more data sets in the pipeline that are due to be published from the TERN Landscapes facility this financial year. So as they come online, they'll be put into that Explorer as well, so that you can, you explore it or use those operations that I described in the presentation to kind of subset it or sample those data sets. I know there are, within the kind of broader TERN repository, that there's quite a lot of other data sets that are already published. I know, I think Guru and Gerhard from TERN Data Services at UQ have been working toward making a TERN-wide, visualizer and explorer. So I imagine the rest of the published datasets, along with the TERN Landscapes ones, will all be commonly available within that when that's finished.
Thank you. Maybe a slightly controversial question for Alison. Can we make the final grant payment from government or ARC or funding source dependent on depositing the data?
You could always try. The history is, of course, I may say, synthesis centres, of which there are a few around the world. The oldest one had a mandated, you must deposit at the end of it. I think they've achieved about 50% success rate. So, and certain NSF, ARC, the eDNA in France have begun to have, at various stages, ARC is quite early, have begun to have that mandated. But who is actually monitoring that regulation? And of course, where? And so this is where I think it's pretty neat that TERN has a bit of a facility going for depositing important data. So having that, means you've got less and less excuse. But we just published a paper on open science, and I love my mother, I love apple pie, an American thing. I agree with open science. Great idea, transparency, all that sort of thing. But actually doing it depends on the tools being available. So I would say it's a bit 50-50 that the government may mandate. It'll certainly help, but it also should invest in the tools and the means, the education, the facilitation to do so. And that doesn't have to happen just within a country. Other countries can help. So I hope that's an answer.
Thank you. Maybe a quick one for Ashley. Would you consider running a workshop or maybe some video tutorials for utilising the datasets?
Yes. So, what we're building at the moment is a set of kind of user guides or quick start guides. So that's going to be the first thing that we roll out. I've kind of aimed to have them complete, for this symposium. So because I imagine people would want to talk about that and want more information. So I can give links to people who ask to those quick start guides and that kind of thing. But if there's enough interest, definitely we could hold workshops or video tutorials as well.
Thank you. How are we going for time, Beryl?
I think we should wrap it up.
Okay. There was one really good question about privacy, but I think hopefully we can address that later in the symposium about how to deal with data privacy for all the disparate contributors. So if you join me in welcoming, congratulating and thanking all our speakers. As Beryl says, you are dismissed.
Well, I find day 2 inspiring. It's been a great day so far, but I think you'd like to go and reflect on some of those talks. So we have morning tea, and if you could be back, please, by 11:15, we would appreciate it. Thank you.